Uh, all we know as the mitochondria that has site for complete oxidations of the foodstuffs in form of all as a monomeric biomolecules like as sugar, fatty acids, and amino acids. And all of them are completely oxidized into CO2, H2O together with productions of 2H. This is called as a reducing equivalent, and this reducing equivalent that pass through the electron transport system, and the electron transport system is located on inner mitochondrial as membrane. And the inner mitochondrial as membrane that we told you last class, because the major content of the inner membrane mitochondria that is a protein in nature, and that has a protein because as a due to the presence of the electron transport system, and there is four complexes: complex first, complex second, complex third, and complex fourth. The complex first is the largest complex, complex second is small, and complex third is large, and complex four is larger. And the complex four that is the that has that is coupled as with the enzyme that is known as ATP synthase. And ATP synthase, there is the real a candidate has to in mitochondria so that accept the energy of proton, and that energy of proton is conserved in form of ATP by phosphorylating the ADP together with inorganic phosphate in form of ATP. Question another point that is how respiratory chain that is arranged on the inner membrane of mitochondria. So the very so first so if we look at the complex as of first complex as of first, that is complex first is known as NADHQ reductase. NADH as a Q reductase. NADHQ reductase. This NADHQ reductase that accept electrons from dehydrogenases that is linked with the NAD. And NAD has to get reduced with receiving the electrons and proton in form of NADH plus HIM. So that if we this also you can see that is the 2H, or this has a 2H that consists of two electrons. Two electrons. This has a two electrons. That is this one, this one, this is, and also two protons. And NAD has receives in such form. This is the minus because it consists of two electrons and one has a protons. And this 2H, that is also this as a proton. And also the electron is accepted by NAD and get reduced to NADH, and finally it has to travel also to the EV quinone, that is also called as quinone. And it get has to reduce into QHSO2. In last class, also we had as explained one kind of inhibitor that is called as rotinone. And this rotinone is insecticides. And this insecticide that is used as for treating the plant for keeping the insect away. But question is, plant also has mitochondria, animal also has mitochondria, insect also has mitochondria, but as a plant remain protected, remain as a safe. The rotinone does not also harm this plant mitochondria, but as it finally damages the insect mitochondria or mitochondria of animals. Because as the electrons flow has a problem, because this NADH has that contain iron sulfur protein and it consists of six 15 polypeptide chain with 6 iron sulfur protein, and this NADH that transfer the electrons to ubiquinone. Then as a rotinone has that inhibit the transfer of electron from NADH, yeah, particularly from iron sulfur protein, that is the FES. This FES, that's why it has to prevent the respiration together with flow of the proton. This is flow of the proton, so that electron and proton both are locked. That's why the inner ATP formation is just a block finally. Or another question is because as it inhibits the electron as a transport system, a respiratory chain at the complex as of first, so that the flow of as a proton, a flow of electron as of from QH2 to successive respiratory chain of complex third, complex as a fourth, and finally the formation of the water is just a block. In case of the succinate, Anand, I am giving Anand. आनंद इधर आ तो स्लाइड कहाँ स्लाइड करते हैं बस वो लोग उसमें था हाँ शेयर किए थे जो अच्छा वहाँ से आ जाएगा हाँ ई वाला हाँ ठीक है इसके बाद दूसरा कैसे लाएंगे अच्छा हाँ से ले लेंगे अच्छा अच्छा ठीक है ठीक आई एम शेयरिंग अच्छा अच्छा ठीक है हाँ ठीक I am sharing these slides that you can see here because as either as a pyruvate or alpha ketoglutarate or malate that is coupled as with the enzyme as a pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase that releases the reducing equivalent, and this reducing equivalent that is further accepted by FMN or FAD, that is known as flavoprotein. From the flavoprotein, the electron is accepted by NAD, and NAD get reduced into NADH plus H ion. And finally has again FMN, that is the vitamin B2 that accept the electron because it is coupled with the iron sulfur protein. And then finally to the ubiquinone. Whenever this one is get inhibited as whenever rotinone is applied as there, a rotinone insecticide or other insecticide that contain barbiturates, and the barbiturate has that is the hydrophobic in nature, and that get penetrated into the mitochondrial membrane. And so this one, the plant that is sprayed with the insecticide, and also they are consumed later by the insects. So then it inhibits the further metabolic processes or oxidative processes of the mitochondria, and also the insect dies in absence of sufficient amount of the productions of the ATP. And so this one has that is the Q, and another complex two has that accept the such electrons from those enzymes that is coupled with the succinate dehydrogenase or acyl coenzyme dehydrogenase, and this dehydrogenase has FAD, and FAD as the cofactor, and this as a cofactor the accept the electrons and then directly transfer to the ubiquinone rather than complex as of first. So that as a succinate dehydrogenase are the substrate that produces the that get oxidations that is catalyzed by the dehydrogenase enzyme, so that has FAD as a cofactor, that then it transfer electrons directly to ubiquinone, and this as a ubiquinone get has reduced FADH2, and so this one then has to transfer electron to complex third and complex as a 4, and finally as oxygen is here, and oxygen accept the electrons, then as protons, and finally it form the H2O. The complex as third you are looking as here, the cytochrome as B1, it also contains as the iron sulfur protein, cytochrome C1 and cytochrome C. That is as it consists of the complex as third. It is the larger as the complex of the mitochondrial membrane. And as the cytochrome C or cytochrome C1 or cytochrome C, it contains the copper atoms. Along with the iron, they also need as they also have the copper. Later, the complex third from the complex third as electrons is passed to the cytochrome A and cytochrome A3 and finally to the oxygen. It also contains the as of copper. One question that is uh, uh, one point to be discussed like the in later past class as I had also discussed that is the newly born has a mammalian baby that contain the has a brown fat in adipocytes. Yeah, brown fat. Brown fat is nothing but it is the adipocyte that contain large number of the mitochondria. Large number of mitochondria means they also have cytochrome system and the cytochrome that has to contain the copper. So that has to it has to absorb the has a wavelength nearly the 450 or 450 that is ranges from the browns. That's why it seems to appear as a brown. That's why it is known as a brown fat. Second one case of this brown fat that mitochondria of the newly born has a baby that has to contain a specialized protein that is called as uncoupler or uncoupler is nothing or that is the thermogenin. Thermogenin is a protein of 37 kd as a protein. It acts as a uncoupler. Uncoupler as a means the during respiratory chain the flow of electron and flow of as a proton gets separated. The flow of electron is continued to the oxygen because oxygen act as a final electron acceptor. So that electron has to continue to has to move on the respiratory chain and finally it is accepted by the ultimate electron acceptor that is oxygen and finally produce the water. But however, it has to uncouple the protons from the electron transport system and so the proton does not have to the move toward the battery inner membranous space for further create the proton motive force so that as a proton motive force is not created whenever thermogenin is there and that's why as the proton motive force that has to not created because proton does not move from matrix to intermembranous space. That's why ATP is not synthesized. That's why as the brown fat that contain the cytochrome system and one has a kind of protein that is uncoupler known as thermogenin and this has a thermogenin that capture uncouple the flow of proton so that energy of the proton that is captured dissipated in form of heat. This one again so that is the respiratory chain of mitochondria. Mitochondrial membrane has transport system. That is also because there is two objective because it's a minor membrane, a matrix of the mitochondria that always maintain electrical gradient or electrical gradient that is the charge. So that if so there is movement of one negatively charged ion, then so there is must be so either inflow of the positive ions or in opposite direction. If there is movement of opposite direction takes place, that is also known as antipode system. In case of the antipode, if also negatively charged enters from to the matrix, then also the one negatively charged also must be exported from the matrix to outside. For example, you are looking as this is the outside outer membrane, either this is the inner membrane or this is the inside that is called as matrix. That's why the matrix that is OH barter splits into OH and H ion. That's why the OH that bear one negative charge it moves from inside matrix to the outer as a membrane. But it also the movement of this OH ion is coupled has with the one movement of one molecule of inorganic as a phosphate from outside to inside to maintain the electrical as a charge because it bears the negative. It is both species are negatively charged species. So that as a movement of the negative charge in opposite as a direction, then as it is also coupled with the movement of the negatively as a charge. Doesn't mean in any case in case of the antipode, if there is movement of positive, then as a positive movement that maintain the electrical gradient. If there is movement of negatively charged substances, either across the membrane, there will be movement of negatively charged ions or vice versa. And यहाँ पर you are second one the pyruvate. Last class because of the pyruvate and as a proton. The pyruvate is produced in the cytosol and it enters the mitochondria for further oxidation, but it bears one negative charge, so that it has the movement of pyruvate is coupled with the proton ions in same directions. This is the same directions. That's why it is called as symport system. That's why mitochondria also has a symporter. An example of this is pyruvate movement of pyruvate from cytosolic sites, cytosol to the matrix of the mitochondria through occurs together with the movement of proton in same direction. This is known as symport. One question is so that is the malate, and malate is produced in matrix of the mitochondria, particularly during the glycolysis. Because during glycolysis, the two molecules of NADH or 2H ion is produced, and cytosol has no respiratory chain, and the mitochondrial membrane is impermeable for the charged particles, particularly NADH plus H ion. So that the NADH or H ion that is accepted by oxaloacetate in presence of malate dehydrogenase, and so the oxaloacetate reduced into the malate. So that as a malate that moves from cytosolic site to matrix of the mitochondria. In return, from the matrix, inorganic phosphate moves in opposite direction toward the cytosolic side, so that electrical gradient has to be maintained. Again, another kind of the example, that is the antipore system. Same thing happens. That is one thing you are looking here, malate and citrate. Because I am this citrate that contains the three molecules of carboxylic acid, and three carbonic acid means it has three negative charge. And also the malate that is produced in the matrix of the mitochondria. And also this is from the matrix of the mitochondria, the malate enters the cytosolic side for further its oxidations, but the citrate that is because we consume lemon or other citrus fruit. The citrus fruit that contain the citric acid, so that citric acid releases its proton and transform into the citrate. And further metabolism of citrate takes place in matrix of the mitochondria. But the entry of the citrate occurs along the mitochondrial matrix, which is compensated by the outflow movement of the malate from the inside, from the matrix of the mitochondria to the cytosolic side. So that the malate has to be a two negative charge because it has two carboxylic anion, and citrate had three anion. So that the citrate has to take up one molecule of a proton, so that it balanced. That is the two electric negative charge that remains here at is two. A malate also bears as a two. So that this movement takes place in opposite direction. It is also called an antipore system. Alpha ketoglutarate and malate. And alpha ketoglutarate is well known, the intermediate compound during the Krebs cycle. However, the glutamate is oxidatively deaminated in cytosolic site. Then it produces the alpha ketoglutarate. And so this alpha ketoglutarate is also the substrate for two amino acids, glutamate and glutamine. But in my cytosolic side, if glutamate or glutamine are oxidatively deaminated, it produces the alpha ketoglutarate. And as further, its oxidation takes place in the matrix of the mitochondria. And so that there's a inflow of the alpha ketoglutarate from the cytosolic side to matrix. Then from the matrix, the malate is exported to the cytosolic side. So that there is two or more examples that maintain the movement of the negatively charged species from the cytosolic side to matrix of the mitochondria, which is compensated by the malate. Last one this one, ADP or ATP. One point to be noted here, because this inner membrane of mitochondria has an enzyme that is called as ADP ATP translocase. This contain, you are looking at the 6 number. This is the enzyme present in the inner membrane of mitochondria known as ADP ATP translocase. So that is because ATP is produced in the matrix of the mitochondria, but it is expenditure in cytosolic side for all the synthetic processes like synthesis of fatty acids, cholesterol, amino acids, and other protein synthesis, and all these processes that need that ATP. Not only in the common cell, but other cell like myocytes, cardiac cells, nephrocytes, neurons, all need the consumptions of the ATP. But ATP is produced mostly in matrix of the mitochondria, and it must be exported to the cytosolic side where there is need of the ATP for metabolic processes or vital processes. And ATP it bears the sum total of 4 negative charge. But the ADP bears sum total of 3 negative charge, and again one molecule of inorganic phosphate. Looking here, the ADP together with one molecule of inorganic phosphate is imported from cytosolic side to the matrix of mitochondria, and the ATP that is produced in the matrix of the mitochondria that is exported. And so this is carried out by an enzyme that is also called ADP ATP translocase. One question for a student is here, because the ADP ATP translocase that also need the energy. But the energy that is needed for ADP ATP translocation are not coupled with the ATP hydrolysis. But however, during the proton flow, later I will discuss that during the proton flow, the formation of one molecule of ADP from ADP and inorganic phosphate, the energy of 4 protons are needed, and out of the 4 proton, the energy of one proton that is consumed for the pumping of the proton, and one energy of 1 protons that is required for the ADP ATP translocation. And this ADP ATP translocation is carried out by enzyme that is called as ADP ATP translocase, and it need energy. But this energy it comes from the energy of the proton rather than ATP hydrolysis. So that point is noted here. Later the energy of 2 proton that is expenditure for the synthesis of ATP from ADP and inorganic phosphate. So that formation of one molecule of high energy rich bond, and it requires the energy of the 2 protons that is produced during the metabolic processes. That's why. Or are there you are looking here, these are the inhibitors. The inhibitor ethanomalimide that inhibit the N-ethyl that inhibit the movement of the OH and inorganic phosphate from so that import of the phosphate from the cytosolic side to the matrix of mitochondria is inhibited. Second, N-ethylmalimide together with hydroxycinnamate that inhibit the import of the inflow of the pyruvate to the matrix of the mitochondria, so that if glycolysis that produces the pyruvate, but further oxidation takes place in mitochondria, or if there is an inhibitor molecules that is N-ethylmalimide, hydroxycinnamate that inhibit the movement of the pyruvate, so that further it inhibit the aerobic respirations or aerobic metabolism. That's why the major more than 95-97% energy that is produced in mitochondria through the oxidative processes, or they have, because they have a respiratory chain that is coupled with the oxidative phosphorylation. If this inhibitor is applying, and then the major production of ATP is inhibited. Last one, that is this question has come in NET once, 2 or more times. Atractyloside, and this atractyloside that block the ADP ATP translocations, so that movement of the ADP from the matrix of the mitochondria to the cytosolic side, and and so that the formation ATP that is produced in matrix of the mitochondria that moves further in the cytosolic side, it get inhibited, so that the again formation of if mitochondria that has actively respiring system, they are producing the reducing equivalent, but however the ADP nucleotide, adenine nucleotide is not also there, then also formation of ATP is inhibited, and further the energy requirement get has that is not available because the mitochondria has been treated as with the inhibitor like reticulocytes. So that is, so this is the inner membrane of mitochondria that has transport system. This transport system that comprises as the movement of OH ion, inorganic as of phosphate, pyruvate, proton together. That is a kind of support system. And also again as the inorganic phosphate as a malate, finally malate citrate and malate alpha ketoglutarate, and last one is the ADP, ATP. That is the on all this one out of these as a transport system in matrix inner membrane of mitochondria, only the ADP or ATP that has enzyme system, enzyme that is the ADP, ATP translocate. And so this one question is there here also. It needs the energy that comes from the energy of the protons. Or okay, later you are also looking here again. This is the ATP, you can imagine that is the ATP synthase, that is the enzyme. Or in last class, also the structure of ATP synthase I had explained, because you know the ATP synthase that has evolved in a prokaryote, and it is okay, and continuously it get conserved, and still it is present in both plant chloroplast and so the mitochondria in the plant cells, and in case of the animal, because so they have eukaryotic cells, they also have only mitochondria. That's why the ATP synthase is present. Second question always also asked during the viva or other, because as the all the nucleotide ATP, CTP, TTP, UTP, GTP, etc., that also have the same phosphoryl potential. But why ATP is known as energy currency of the cell? If you look at the during the evolutionary as a processes, so only as a single enzyme ATP synthase got conserved, and as moves from prokaryote to the higher eukaryotes that has contain the ATP synthase, and as it capture the energy of the protons and further phosphorate the ADP to the into the ATP, that is also called as energy currency of the cell. It was the Lippmann and also who proposed the structure of the ATP, and he was also awarded with the Nobel Prize. He also gave the symbol for the energy, energy, high energy rich bond. I will discuss later. So this one is you are looking at, so this one, this is the respiratory has a chain, a respiratory complex, that is complex first, second, third, and fourth. Together with that is the ubiquinone. Complex has a first that is that linked with the NADH reductase, succinate has a complex second, succinate reductase, third cytochrome C reductase, and last one has only one enzyme in the respiratory chain of the mitochondria, that has oxidase enzyme. And this oxidase, you are looking here, but this is the outer inner membrane space of the mitochondria. This one is the matrix of the mitochondria. During as oxidative as processes, this 2H is produced, and 2H that consists of the electrons and the protons. So that as electron moves on as this respiratory as chain together with accumulating the protons, and also the proton moves from matrix to the inner membrane space through three as complexes. Complex as first, complex third, complex fourth. The complex as second that doesn't have the proton flow system. That's why the proton flow has from the matrix to the inner membrane space where it get accumulated. And accumulation of the proton, because the one hypothesis regarding the oxidative phosphorylation of mitochondria, it was the Peter Mitchell in 1961 who has proposed, because as before him there was a lot of the question how mitochondria energy has got conserved during the oxidative processes or respiratory processes. So then it was Peter Mitchell in 1961, and also Lehinger, later one Lehinger also made their contribution in energy conservation in during aerobic respiration. So Peter Mitchell, who proposed the hypothesis that is known as osmotic hypothesis, and this osmotic hypothesis that is that explains the flow of the proton from the matrix to the inner membranous space, and as it create and accumulations of the proton in inner membranous space that develop accumulated as a proton that create the proton motive force, and this proton motive as a force that further allow the movement of the proton from the inner membranous space to the matrix. But as this movement also takes place, the ATP synthase enzyme. This is the ATP synthase enzyme, because this one consists of the entire enzyme consists of the 9 polypeptide chain, and this is the knob that faces in as toward the matrix, and also the proton has a channel or a stalk, that the proton channel that faces to the inner membranous space, and also the stalk has that traverses the inner membrane of the mitochondria. Then as the inner membrane a space of the proton, because nearly what has 10 protons, and that is if the movement of the proton from matrix to the inner membrane space, there is a movement of 10 protons takes place, as reducing equivalent as 2H is produced during the dehydrogenase enzyme. And the 10 has a proton, it means 1 pH has a decrease. So that the matrix of the mitochondria, as when proton moves from matrix to the inner membrane space, it means the pH of the matrix of the mitochondria increases because proton is decreased, and also here the proton concentration increases, and as a proton concentration increases, it means pH has to decrease. So that also, and 1 pH is equivalent to the at least also 10 protons. So that there is a decrease and there is a difference across the mitochondrial membrane is about 1 pH. Or so that as the inner matrix that become alkaline, or whereas as the inner membrane space that become acidic. But anyway, the differences in as of one as a proton, that is the electropotential electrochemical gradient. This electrochemical gradient due to the movement of the proton from matrix to the inner membrane space that generate energy about 1.14 kilocalorie energy. Yeah, sometimes 1.4 kilo, not 1.14, 1.4 kilocalorie energy. So that if there is any difference, or secondly, in case of the volt, it is nearly what 0.14 as a volt. So this as a 0.1 volt. If you put it into the formula delta G, then as a RT log Faraday, because it contains the charged particles, and as a Faraday electron membrane potential, and as pH gradient. So then as the energy equivalent as becomes to 224, 0.224 kilocalorie energy, and this 0.224 kilocalorie energy that is equal to 10 protons. And so this also 10 also protons, because if you have to divide the 2 to 4 by also 10 protons, then also it becomes equal to 5.24 energy as for one proton. So that for synthesis of ATP from ADP and inorganic phosphate, the requirement of the has of 4 protons. The has of 4 energy of the 4 protons. This one, because as of the it means as of this proton, this proton, this proton, this proton. There is just 4 protons, and also the protons energy of the one as a proton, that is expenditure during the ADP ATP translocate system, translocate enzyme, and also the energy of the SO2 has a proton. The energy of the 2 proton, that is equivalent to 10 kilocalorie energy, and this 10 kilocalorie energy has that is expenditure to conserve the energy in form of high energy rich bond by capturing the phosphorylating the ADP together with inorganic as a phosphate in form of ATP. So that if ATP hydrolysis takes place, then it liberates the minus 7.3 kilocalorie energy. So that has this as energy of the two protons is required as for the capturing phosphorylations of the ATP in together with inorganic phosphate to form ATP. And as the energy of the one has a proton. The energy of one proton, that is expenditure as for the proton has a pump, because and a proton flow, because it is contain delta epsilon, gamma delta epsilon. This is the hydrophobic in nature. It remains embedded in the inner membrane of the mitochondria. So that is the movement of the proton as from this inner membrane space to the matrix, that is coupled as with the synthesis of ADP with inorganic phosphate into ATP. And so later as the electrons and this proton and is further combined to form the electron is finally accepted by the oxygen to produce the H2O. So that this is the in ATP synthase enzyme, that is coupled also with the flow of a movement of a respiratory chain that consists of the movement of the electrons and also protons. Another one, that is this one, that is also because also the mitochondria so many kinds of inhibitor. Here as we are explaining somewhere, that is the uncoupler. Second one is the inhibitor. An inhibitor is of two kinds. One is inhibitor is the inhibitors of the respiratory chain. Another has the inhibitors of the entire electron transport system. And so that is in case of the uncoupler like oligomycin or also the thermogenin has a protein, uncoupler has so that uncouples the movement of the protons. So that is the movement of the proton from the matrix, from the inner membrane space to the mitochondria, because the oligomycin that is present in a stalk of the elementary particle. That's why it is also known as oligomycin sensitivity conferring as a protein. The oligomycin sensitivity conferring as a protein that inhibit the flow of the proton from the inner membrane to the matrix through the ATP synthase enzyme. That's why electron has a flow continuously as from complex first to the final acceptor like oxygen. But however, movement of the proton is inhibited. That's why ATP barter is produced, but ATP is not produced. That is known as uncoupler. Second one, that is the inhibitor of the entire respiratory chain. Just for that is the movement of the electrons and protons also. Common example is that is the pyridine A, ambobarbital, and rotein. All has these three group belongs to barbiturate or carboxyl TTFA. That these are inhibitors that inhibit the movement of the electrons from the iron sulfur protein to the ubiquinone. So that as electron movement is a block. That's why it inhibits both further as a movement of the electron and also the further movement of the proton. So that it inhibits the entire formations of the ATP. That's why the animals that receive this kind of insecticide, or even though someone has to consume the insecticides during because they tend to commit suicide, then also the respiratory chain is just a block, slowly as a block, because it inhibits the movement of electrons from a complex first to the ubiquinone, or whenever through the along with the blood vessels, then also along with the blood vessel it moves to the cardiac cells or as brain cells, neuron cells. Then it is sensitive also to this kind of the insecticide, and also the potentiality of this insecticide or concentration of insecticide that increase in large amount, that then also people also die in sometimes, or if its concentration is low, then also their survival may be chances as for after treatment, but the possibility is negligible. In as a complex as a third, again as you are letting the uncoupler, and so this as uncoupler again oligomycin or the antimycin A, and this antimycin A has that has blocked the movement of the complex as a third from the cytochrome C to C1 to cytochrome C, and Finally, then to cytochrome A and cytochrome A3. Because this anti-acomycin, that is the anti-acobiotic that inhibit the movement of the flow of the electrons and the protons. That's why it inhibits the both formation of water as well as ATP formation. This one you are looking at, so this is the this H2S, hydrogen sulfide, carbon monoxide or cyanide group. And so this comes sometimes cyanide has a group that is a potassium cyanide, that is the highly sensitive potential intoxicant or poison that has that kill the organism, animals including human beings in short period of times, because this cyanide that inhibit the cytochrome A the complex as a 4, particularly that has an enzyme known as ATP synthase. So that it has blocked the both flow of the movement, the electron acceptor, that is the oxygen, yeah, inhibitor that inhibit the electron flow from cytochrome system to oxygen, and during which the movement of the proton is also as a block, so that the disperatase chain is as a block. Then hydrogen sulfide or the hydrogen sulfide, so that is the in case of the agriculture as a field, or that is a contain as the sulfur, and the sulfur react with the water to form hydrogen sulfide, and this hydrogen sulfide that is the very harmful to the paddy plants or aquatic plants, because it inhibits the absorptions of the oxygen or consumptions of the oxygen as an ultimate electron transport system. But in case of the paddy fields or other aquatic body that has received the hydrogen sulfide through the industrial effluent, so then the sulfur bacteria, numbers of the sulfur bacteria is also there. One kind sulfur bacteria like Begiatoa, that is the chemosynthetic bacteria, autosynthetic bacteria. They are autotrophs, but so they don't use the water as an electron producers. That's why they use the hydrogen sulfide as an electron, source of electron for the reductions of the carbon in form of glucose. So that's why the air aquatic as a body that has iron sulfur received as effluents from industrial and other processes. But so this if they have an sulfur bacteria, Begiatoa and others, so it means also they protect the other organism by consuming the hydrogen sulfide also. Well known as the pollutants, that is the carbon monoxide, because carbon monoxide is produced by incomplete burning of carbon dioxide, or in you can say insufficient of oxygen amount of the oxygen is here or there. So then the carbon containing the fuel or carbon containing the organic compound that incompletely as a burn, and as the produces the carbon monoxide, and it inhibits not only to the respiratory chain as it enters the mitochondria, because it is permeable. Permeability of the carbon monoxide is just more than that of the carbon dioxide and oxygen also. Second objective is that in case of human beings, there is a carbon monoxide that is inhaled during the breathing process or respiratory processes. So then as the carbon monoxide so that sits, yeah, that combines with the hemoglobin, and so this together with the hemoglobin, it binds with the porphyrin ring to the nitrogen atom, and so that and it interacts with the nitrogen atom through by forming an electrostatic as a bond, and so that they make it make the stable as a compound and inhibit further as a transport or carry transport of the oxygen from lungs to the other part of the tissue, so that it is more as a harmful as the gases. Carbon monoxide, if it is inhaled during the respiration, or if it is carbon monoxide that is also produced by the burn out of the RBC, burnout of the RBC that also produces the carbon monoxides in during metabolic activity. If the carbon dioxide is not removed, because as it is removed in so many processes, but for the immediate process, that is carbon monoxide that combines with one molecule of oxygen, then it produces CO2, so that CO2 is less harmful than that of the carbon monoxide. So that these are the inhibitors of molecules. All these are. That is the malonate. And also this as a malonate, it is called as inhibitor, or also this is known as, because uh, the malonate also that resembles with the succinate. So that also the enzyme used to catalyze the succinate into fumarate, that is the succinate dehydrogenase. So that also the malonate act as a competitive inhibitor. Malonate is act as a competitive inhibitor, so that this as a malonate both are resemble, but as it contains as a 4 carbon, it contains as 2 carbon more. So that as the malonate that has to actively bind the in the active site of succinate has to dehydrogenase enzyme, so that as it inhibit the succinate dehydrogenase to for further oxidation of succinate. That's why it inhibits of the both FAD, flavinin, adenine dinucleotide, and RN sulfur protein. That is it is coupled also with the proteins, so that inhibit, and as electron doesn't move from this, then also the electron from FAD or complex 2 to we cannot get inhibited, like as a carboxyl TTFA. That's why ATP synthesis is also as a blocked. This one now, this is the basic compositions of the respiratory HSO chain. And as this respiratory chain, that is the number. This one you are looking here, I am telling here, because also there is complex first, complex second, complex third, and complex also 4. And also it is the mass in kilodalton, but also this is the coupled also with the genes that is present in the mitochondria. And last class also we had explained the mitochondria has covalently closed circular DNA, and the covalently closed circular DNA that consists of more than 16,000 nucleotides, 16,500 nearly approximately. And so out of that, this nucleotide is clustered in 37 genes, and this also 37 genes, out of which 24 are essential for synthesizing the transfer RNA or ribosomal RNA, but 13 genes. This 13 genes that conjugate with these enzymes like NADH dehydrogenase, and then complex third and complex 4. Succinate doesn't have. So that is this 13 has the NADH dehydrogenase that has contains 7. The polypeptide chain of complex first, that comes from the mitochondrial gene, and rest 14, if you subtract it from 43, then you will get the 37. The 37 polypeptide chains are encoded or produced on the nuclear genome, so that it is a kind of the symbiotic synthesis of the this enzyme complex first, because it's the functioning of the NADH dehydrogenase or synthesis of NADH dehydrogenase enzyme that require the genome of both nuclear genome as well as mitochondrial genome, so that both are interdependent. And in that interdependency, 37 polypeptide chain that is synthesized on the nuclear genome, and so the 7 polypeptide chain are synthesized on the mitochondrial genome. Complex succinate dehydrogenase that has to contain one polypeptide chain, however doesn't. But as the main has the case of succinate, complex also third, that is also that contain, that is required for the synthesis of 3 polypeptide chain. 3 polypeptide chain you can say, that is synthesized on the nuclear genome, and rest of the polypeptide chain are encoded or synthesized on the nuclear genome, and also 3 polypeptide chains are on the mitochondrial genome. And the cytochrome oxidase, it requires 2 polypeptide chain and 1 polypeptide chain that is synthesized in the ATP synthase enzyme. So that is 7, 3, 2 or 1. Total 13 polypeptide chain are synthesized on the mitochondrial genome. Question 1 is, because if you look at the history of the mitochondria, the oldest mitochondria that has been studied in human case, because in African peoples, that is the oldest tribal or ethnic peoples of the South Africa, that is also known as pygmies. and pigmeat is known to oldest human race. And in case of the India, that is Dravidian, that is known to be as the oldest as a race. So that also the mitochondria of also these peoples were also compared, or the mitochondria of the pygmy peoples also compared with the other as peoples in India across the some in different states of peoples that they have collected as samples were collected from different sources in other state also. The entire as a country in India, the genome sequences was very similar in between Dravidian peoples and pygmy peoples. So that it is known as the human race very first of all evolved in South Africa, and from the Africa they migrated to other as a part of their country, and along with the heat zones, and they moved toward the polar regions, a cold climate, temperate climate, or hot climate, or tropical as a climate. Slowly as so their physiological as a process change, a skin color as a get as a got changed. That's why. And second one, this is the because also one point also comes from the genome of the mitochondria. It has been started as so that there is a lot of the mutation have been taken as a place in as so the mitochondrial genome of the humans. But however, the complete evidences are still required to for the debate. But in case of the NADH dehydrogenase, it consists of the 7 polypeptide chain, and it's also the fourth polypeptide chain that is known as ND4. That is known as ND4. NDSO4 means because the enzyme has that is linked with the dehydrogenase has that is linked with the NAD, that contain the NAD as a prosthetic has a group. So that the fourth polypeptide chain that has that contain the arginine, or if the arginine is also substituted by the histidine, same both are the positively charged, and but also uh, there is a mutation in the mitochondrial genome, so then also the ND4 has to get affected, particularly of the fourth polypeptide chain of the NADH dehydrogenase as enzyme, or complex as of first as enzyme, that is NAD that contain the NADHQ reductase. So then also arginine is substituted by the histidine. So then it leads to a disease that is called as epileptic or as a myocolic as a disease, in which as muscles and as a brain cells are badly affected because productions of the ATP does not take place in sufficient amount to as a continue the as a processes. So that as the it is known as a kind of extra chromosomal inheritance, because already know the mother that invades the mitochondria in all kind of progeny, male progeny as well as female as a progeny. So that mother mitochondria that is essential as a for uh, maintaining the body physiology, particularly for the energy transformation. If there is any mutation takes place, then also the 4 or 5 kinds of diseases that has been studied from the mitochondrial genome. So many muscle dystrophy and others, the energy that has muscles, the body has a part that need has more energy during the locomotion, contractions, heart heartbeat, and so many so kinds of the body that need consumptions of the more energy, or if the energy is not produced in sufficient amount because the respiratory chain is become defective due to the mutations in the mitochondrial genome. So that's why the individual that receive the defective genome from the mitochondria have uh, so many kinds of problem, particularly as for the energy as transformation, energy productions, etc. This one, this is the simply as Anand. This Now, so you are looking here. You complex as of first, complex as of complex third, and complex as of fourth. Why as a question is why complex first, complex third, and complex as of fourth? During through this as a complexity, if as a movement of proton takes place, so then as the three ATP is produced. If as the reducing equivalent is accepted as by the NAD and it gets reduced into NADH and HIN, then it generates the three molecules of ATP. But if as of their complex as a second that accept the as of protons and like as a from oxidation of succinate to fumarate, it only generate the two molecules of ATP. And as a here as a, you are looking as a, that is the sum total of four protons. And as a, the four as a protons, this one is four proton and this is the 2H proton. The sum total of as that is 4, and 2. 10 has a protons. And so this has a 10 protons are exported from the matrix to the inner membrane of mitochondria. That is as you are looking, that is the P side. P stands for 2. P side, that is the proton, or another one, that is the P for positive side. Because the proton has from the matrix is exported to the inner membrane space across the mitochondrial membrane through F0, F1 as a complex. So then as a negate inside, that becomes more and more negative, and as a outer one that becomes as a positive, so that it creates the electrochemical as a gradient. That consists of the two kinds of the gradient: electrical gradient, that is the negatively as a charge, and also that is the here one is that is the positively charged. Second one, that is has this proton, that is another has a meaning, because it very first it contributes the electrical gradient by so altering the electrical charge, or second one, that is the pH. So that is the 10 protons from the matrix and transported to the inner membrane space, that then it reduces the 1 as a pH in inner membrane space of the mitochondria, and matrix that has become slightly more alkaline in comparison to inner membrane space. And so this proton as that has energy. It contains the energy of this fuel. And so the energy of this fuel in form of this proton must be as captured for the ATP synthesis, that is known as vital energy. And so this as a vital energy of the four protons that is captured by enzyme that is known as ATP synthase. And this ATP synthase in alpha 3 or beta 3, out of the alpha 3 and beta 3, the beta has a 3 that has energy capturing ability, because it consists of beta empty, beta ADP, or beta ATP, or alpha beta empty that accept the ADP and as inorganic phosphate, and then as it transform into the beta ADP and inorganic phosphate, and its phosphorylation, that energy that is provided by the protons. So that energy of this proton is conserved in form of ATP. So that as this ATP is also produced, so that beta subunit transform into the ATP, and this ATP as a ADP nucleotide continue to move to the ATP synthase enzyme. Then ATP is further released, and this ATP is released. That this ATP that is found in matrix of the mitochondria must be exported for the cytosol for expenditure, because if there is ATP nucleotide, all the adenine nucleotide exist in matrix of the mitochondria in form of ATP. So that so further it inhibit the movement, aerobic electron transport system or oxidative phosphorylation, because there is no more ADP as a molecule. So that the synthesis of the ATP that depends upon the availability of the ADP inorganic phosphate, and the energy of the protons also. But ATP concentration of ATP in matrix is higher, then it has itself respiratory chain or electron transport system or proton flow all are inhibited. So that the ratio of oxygen, phosphorus oblique oxygen, this is the phosphorus, this is the phosphorus, and also this is the ox ratio. that the ratio of the so phosphorus and process that is equivalent to electron depends upon the two electrons, but the electrons, but that is the 2H. Or if ATP is present or it is not expenditure, so then as a further automatically so it is just a block this one. Or simultaneously, if there is higher concentration of oxygen in the mitochondria, so then how mitochondria has to get as protected or cell get as protected, because lipid as a membrane of the plasma membrane or as a cell membrane or cell organelle as a membrane that is much sensitive to oxygens. So that is this oxygen that damages the membrane system of the cell organelle or cell. That's why the cell contain the lysosome. That lysosome that is perform slowly the sometimes it engulf the mitochondria by making its membrane surrounding this mitochondrial membrane, or engulf it that is through the process known as autophagy. So that my lysosome also has another system to protect the cells against the oxygens or other other oxygen species, that then has to mitochondria cell has to protect itself due to the autophagy process of the lysosome. The mitochondria has another system that is known as cytochrome P450 oxygenase. The cytochrome P450 oxygenase, this is the enzyme that is required for the synthesis of the cholesterol. So that is this one cytochrome P450 that contain the present in the membrane of the mitochondria, that is also coupled with the flow of the electron, because the cytochrome P450 is simply stand for because the cytochrome that absorb the wavelength of the 450 nanometer. That's why it got the name cytochrome P450. And synthesis of the cholesterol has that need NADPH. So that, and as the mitochondria that is concerned as with the producing the NADH. But as concentration of higher NADH occurs in matrix of the mitochondria during the oxidative process, then or adenine nucleotide is not also there because ATP has already produced or concentration of ATP has already increased. So then also the membrane of the mitochondria so that has cytochrome P450 that accept the electrons from NADH and it deliver this electron to the NADP, and NADP has to get reduced into the NADPH. That's why so all the reductive pathway like as a fatty acid synthesis or fatty acid synthesis, cholesterol synthesis, steroid biosynthesis, all the sex hormone, glucocorticoids, mineralocorticoids, all as these hormone or cholesterol molecule like as the bile, etc. all also needs the NADPH for its synthesis as a because that act as a reducing equivalent or as the donor of the 2H ion 2H. So that as a mitochondria has during aerobic respiration, aerobic processes that generate more and more as a 2H, and this 2H is accepted by NAD and get reduced into the NADH plus H ion. Or if it has a higher concentration of ATP, then NADH plus H ion that transfer its a reducing equivalent to its counterpart like as NADP, and NADP get reduced into NADPH and H ion that is further exported, further utilized for the synthesis of the steroid hormones or cholesterol. Steroid hormones, not cholesterol proper, because as a cholesterol is being synthesized in the cytosolic site, but as a few hormones, the steroid hormones are synthesized in the mitochondria. The one as a process is also here. Second, also the one question, in case of the as a plant, ये कैसा हटेगा बेटा? In case of plant one, आनंद, आनंद, अच्छा यहाँ पर